Good afternoon, ladies and gentlemen. I'm Bradley Flett from the ARC Grain Crops Institute in Potchefstroom, and the title of my talk today is The Influence of Cropping Systems on the Incidence of Maize Ear Rots and Mycotoxins in Maize Grain. Just to give an introduction to the topic, as you know, maize is an important staple food, feed and energy crop in South Africa. Crop production systems conserving soil moisture and topsoil preservation are widely adapted to production areas in South Africa. Conservation agriculture, or CA as it's known, is a sustainable practice that branches from minimal soil disturbance, cover crop retention, and crop rotations. The benefits of CA include increased fertility, water infiltration, prevention of soil erosion, and it's more economical. No-till and cover crop retention have been linked to high disease incidences in certain areas, certain cases. Predominant maize ear rot pathogens generally overwinter on crop residues. Fusarium ear rots caused by Fusarium verticilloides, Gibberella ear rots caused by the Fusarium graminearum species complex, and then Diplodia ear rots, which is caused by Stenocarpalomydis. Maize ear rots are accountable for substantial yield and quality losses in maize grain. Upon infection, maize ear rot fungi may produce toxic metabolites, which are referred to as mycotoxins. These include the fumonisins, which affect the liver and cause esophageal cancer, deoxynivalenol or nivalenol, which results in vomiting, feed refusal, and weight gain suppression. Zearalenone is a estrogenic mycotoxin, and it leads to reproduction problems, particularly in pig farms. Limited control options for maize ear rots and mycotoxin accumulation exist. It is therefore important to investigate agricultural practices and their role in disease incidence and mycotoxin contamination. So materials and methods, as you can see here, we got a number of treatments for the 5 years trials at Buffelsvallei. These were maize monoculture, conventionally tilled, maize monoculture under no-till, and then rotations with cowpeas and sunflowers, and then a rotation with babala, cowpeas, and sunflowers. But these are all done under no-till. So each year there was at least one of each treatment to sample. Then similarly at Erfdeel, which is on a sandier soil, there were similar crop rotations, just we did not include the sunflower. The 2 middle rows of each treatment block were harvested annually from 2009-10 to 2014-15. Visual ratings were done for Fusarium and Gibberella ear rots and for Diplodia counts. Samples were then threshed, milled, and subsamples of 250 grams were used. Samples were then subjected to DNA extraction, a cryogen plant minikit to quantify the biomass of the fungus. Mycotoxin contamination was done using HPLC, and data was analyzed using ANOVA. Skew data was log transformed, and Fisher's protected least significant difference was calculated at a 5% significance level. The results showed there were no significant effects of cropping systems on disease incident and mycotoxin contamination at Afdeel, which is the sandy soil, if you could remember. Cropping systems affected disease incidence and mycotoxin synthesis in only 3 years of the 6-year study in Buffelsvallei. No significant differences in F. verticilloides, biomass concentration, cerealinone, and nivalenol were recorded across all treatments and all seasons. At Buffelsvallei 2010-2011, you could see the fumonisin levels were significantly higher in the sunflower maize treatment, and they were the lowest in the cowpea maize and the monoculture conservation tillage. But these did not differ significantly from each other. All the other treatments differed significantly from the sunflower maize crop rotation. At Buffelsvallei in 2010-11, the mean Fusarium graminearum target DNA was quantified, and you can see that the significant treatment was Babala cowpea maize, which had higher target DNA than any of the other rotation or monoculture treatments. This doesn't totally make sense. One would expect the Babala cowpea maize to be either insignificant or lower than the monoculture maize. At Buffelsvallei in 2011-12, the mean deoxynivalenol levels during the 2011-12 season was significantly higher in the monoculture maize conservation tillage. Just conventional tillage was higher than all the other treatments. This would stand to reason that, and one would expect it under the normal tillage systems. Mean diplodia in maize grown under different tillage rotation systems were low under Babala maize, Babala cowpea maize, and Babala sunflower maize. However, these were significantly lower than any of the monoculture maize treatments, the cowpea maize, the sunflower maize, which shows that a 1-year rotation away from maize is not as efficient as a 2-year rotation away from maize. In conclusion, there was variation in the effects of cropping systems on the disease incidence and mycotoxin contamination across all the seasons studied. Significant effects of the crop rotations on Fusarium graminearum infection, fumonisin accumulation were evident at Buffelsvallei only in 1 season each. Significant effects on tillage and diplodia infection and DON accumulation was also evident. These results do support findings that maize monoculture leads to a buildup of diplodia inoculum. This was found by Flett 1991 and other researchers as well, and they elevated DON levels. The enhanced disease and mycotoxin accumulation in rotation with some crops can be attributed to the ability of Maize Ear Rot fungi to persist in maize residues for longer periods. This study contributes to the understanding of the effect of conservation agricultural practices on Maize Ear Rots and their resultant mycotoxins in maize. I'd just like to acknowledge the University of Stellenbosch, the Maize Trust, ARC, and the NRF for funding for this project. Thank you.